Welcome to the latest mini pharmaphorum podcast. In this episode, Simon Powell, the CEO of Ribonexus, agreed to join me in London after our plans to do so at Bio Europe Spring in Basel fell through. Here, we discuss the 2021 founded company's recent work and how it's on the cusp of identifying its first drug candidate. Put together from long-term research programs at Gustav Roussy, Institut Curie, the University of Strasbourg and Pierre Fabre, our conversation ranges from more lately well-known mRNA to the possibilities of EIF4A. Join us on this discursive oncological and developmental journey, and as ever, thanks for listening. This is Nicole Raleigh, web editor for pharmaphorum, and I have today with me for this episode of the mini podcast, Simon Powell, CEO of Ribonexus. So before we delve into things, Stephen, can you perhaps tell me what Ribonexus does and what you yourself do at Ribonexus?
So I am the CEO of Ribonexus. We are a new company which has been put together to really develop and ultimately commercialise very long-standing research programs from 4 different sources. Our primary goal is to develop drugs that overcome tumour resistance. That is, tumour resistance to existing drug treatments.
Yes, when you mentioned these 4 groups, I know there are 3 world-renowned French research institutes: Gustav Roussy, Institut Curie, and University of Strasbourg. The 4th one is?
So the 4th one, which develops some of the core intellectual property, is the French pharmaceutical company Pierre Fabre.
Mm-hmm.
And so the way that they've come together, Gustav Roussy is very much a clinical centre of excellence in cancer. Mm-hmm. And so they bring very good clinical oversight to what we're doing. The Institut Curie are particularly interested in the mechanisms which underpin some of our drug approaches. The University of Strasbourg has particular strength in medicinal chemistry, so creating novel chemical structures. And indeed, Pierre Fabre had been working in this particular field that we're going to go on and talk about. And therefore, they laid down some of the original intellectual property and created a library of potential drug compounds.
So to focus on Ribonexus, originally, apparently you weren't called Ribonexus, you were called Aglea Therapeutics.
That's correct, yes.
Because it is a very new company itself, Ribonexus, November 2021.
That's correct,
right?
yes.
So in April 2022, Ribonexus received €2 million in deep tech financing from BPI France. And so I just want to sort of know what's been happening since then, since that funding.
So the overall funding picture is that the founding investors, which includes Pierre Fabre, the French pharmaceutical company, and also 2 financial investors, they had committed back in 2021 €4 million into the company, invested as equity. And then indeed we received an additional 2 million from BPI France. What that has enabled us to do is obviously establish the company and start to pursue the business model that we are pursuing. But of course, underneath that, really start to move forward discovery programs in oncology and take the original intellectual property and move it forward so that we can progress to identifying at least one drug candidate, which we are in the process of finalising at the moment. So from inception to date, we've established a larger library of potential compounds. We've been testing those compounds en masse, both in vitro and also in vivo models. Mm-hmm. And we are on track to select a candidate for formal preclinical development within this quarter, in fact, so quarter two 2023. So the real discovery phase of the company has lasted almost 2 years. But of course, the science that underpins what we're doing from the three French institutes plus Pierre Fabre goes back way earlier than that.
How early?
At least 10 years.
Okay.
So there's a huge amount of scientific knowledge which has been endowed into what is now Ribonexus.
Okay, so this 2 years of discovery and development, this has been of both small molecules and biological therapeutic products.
Only small molecules.
Only small molecules, okay.
Yes.
Obviously in the field of oncology, but I did look on the website and it mentioned other disease fields. Is this a future plan or is this something at the moment?
No, that's something we've embarked on since my arrival.
Okay.
So what we have now done is put in place the foundations of a pipeline of activity which encompasses 3 programs. 2 of those programs are in oncology and are focused on the origins of Ribonexus in targeting a very specific cellular mechanism, which we may come on and talk about. But the chemical class of compound that we're working on, which has its origins in plants in the family called Aglaia, hence that original name. From those natural products, we have created this library of synthetic analogues. So we've produced a variety of different compounds from those natural products, all of which can be manufactured synthetically. And these compounds have been demonstrated by some of our founding scientists, both at Gustav Roussy and also University of Strasbourg. They have been shown to be effective not just in inhibiting cancerous tumour growth, but also they've been shown to be effective as potential antiviral agents, and also they have anti-inflammatory properties. So what we have been doing over the course of the past 6 to 9 months is indeed establishing 2 oncology programs, a lead program and a follow-on program, but also looking at a 3rd program to really test our compounds in an inflammatory setting. So we're talking about acute inflammatory conditions such as colitis or chronic conditions such as arthritis. But that's very much encapsulated in that 3rd discovery stage program. So 2 oncology programs and an anti-inflammatory program, all based around the same chemical class of compounds that are called flavoglines, which are, as I said, have their origins in the plant family Aglaia.
That's fascinating, what you've just been describing with the plant family.
We have to be quite careful because the more that we dig into the chemistry of these compounds, the more complex the mechanisms, but also we have to stop ourselves getting carried away, because if you go through the literature, the scientific literature, you find that they have some efficacy in a whole variety of different disease states. So have to be careful that we don't present it as a super drug. But certainly it's, from our purposes, we're focusing very much on oncology and inflammation.
Okay, so if we focus on the oncology for now.
Yes.
You're essentially looking at overcoming chemo resistance and restoring sensitivity to targeted therapies. And it seems that one solution you found is disrupting a protein synthesis downstream of the typically targeted metabolic pathways. Could you tell me more about that?
Yes, so cancer cells are proliferating, growing very, very quickly. Therefore, they have an enormous appetite for a number of metabolic processes, one of which is protein synthesis. So they have a very rapid turnover and requirement to generate new proteins, additional proteins. And what has been identified is that a lot of the signaling pathways, which have been the target for newer cancer drugs in a whole variety of different areas, those signaling pathways converge on a mechanism which is central to protein synthesis. So this is typically upregulated in cancer cells. So there's a high level of protein synthesis ongoing. Now, if you target one of the pathways that converge onto protein synthesis, cancer cells are very smart and they can actually work around, they can bypass that inhibition if you're targeting one particular signaling pathway. But but the majority of the signaling pathways, the oncogenic signaling pathways, are converging on the protein synthesis mechanism, then if you inhibit that protein synthesis mechanism selectively, because of course protein synthesis also takes place at a lower level in normal, healthy cells. So if you can target that upregulated protein synthesis and inhibit protein synthesis in cancer cells, you kill the cancer cell. Ultimately, you inhibit tumour growth. But there isn't really a way round it for the cancer cell either. So what we are looking at in particular is to develop drugs that target that protein synthesis mechanism, but can be used in combination with the newer pathway targeting drugs as well. So we're not saying that our drug will supersede those drugs, but in some cases, 50% of patients may well be resistant to pathway targeting drugs. So we're particularly interested on helping those drugs be more effective and to eat into that resistant population.
Okay, so if we dive down deeper now, it seems that this inhibition you're going after has to do with something called, and I may very well say this wrong, a eukaryotic translation initiation factor 4F, otherwise known as EIF4F, is that correct?
Correct.
Are you able to tell me more about that, or is that as much as you want to?
No, I can tell you just why. We can probably drop down one more step.
One more level, yeah. One
more level without getting super complex.
Yes.
But the key steps in protein synthesis generally are that mRNA is formed as the cell reads from its DNA. mRNA then travels carrying the message for the structure of the protein in question. mRNA moves towards intracellular structures called ribosomes. The ribosomes read the code which is in the mRNA and translate it ultimately into a protein. So that translation step happens when the messenger RNA, which we're all very familiar with now because messenger RNA has been one of the key components of COVID vaccines. But the messenger RNA indeed carries the message, which is then translated at the ribosome and ultimately forms a protein. Now, there are various components of the EIF4F complex, one of which is the one that we are particularly interested in, which is 4A. So it's EIF4A. So we're very interested in this subcomponent. And what happens is that this subcomponent is responsible for latching the mRNA into the ribosome so it can be read effectively. And in particular, the mRNAs that are carrying the messages for oncoproteins, so cancer proteins, have a particular structure which needs to be unraveled. And EIF4A, which is a component of EIF4F, unravels this structure. So if you inhibit EIF4A, you stop the reading of the mRNA and you stop, therefore, protein synthesis. So we are targeting a very specific component and a very specific cellular mechanism. And our founding scientists have demonstrated that in resistant cancer cells, these EIF complexes are upregulated, so they have a much higher level of activity. So if you inhibit them, then you overcome that resistance. And that has been demonstrated now in vivo by our founding scientist group from the Gustav Roussy, Professor Caroline Robert. She, together with the group at the Institut Curie, have demonstrated that if you knock down that activity by inhibiting that component, you can overcome resistance.
Okay, so when you say about this demonstration, are you able to tell me more about the trial which demonstrated that?
It was done in vivo, so it's been done in experimental models, animal models.
Yeah.
But it has demonstrated that in those models you have tumours which are resistant to a pathway inhibitor called a MEK inhibitor. And if you then co-administer the MEK inhibitor with an inhibitor of EIF4A, you have a synergy between the two, which results in inhibition of tumour growth. And we have subsequently now, as Ribonexus, expanded that concept, looking at our lead compounds and looking at how they combine, not just with MEK inhibitors, but other pathway inhibitors, and indeed in a variety of different tumour models.
Can you tell me more about those tumour models at all?
So we haven't settled on a final lead indication, but we're not far away from doing so. But almost certainly, given our scientific heritage, we will be continuing to work in melanoma. We're also looking at non-small cell lung cancer, which is a very significant, poorly met medical need. And we may well also look at pancreatic cancer.
Yes, that's definitely, although we're all valid and worthy, that's one of the cancers which certainly has more space for research. So to put it into sort of the real world terms, all this 50% of patients who are resistant to current therapies, what does this mean for them?
So the overall objective of what we're doing is to enable patients to live longer. So we would hope to be able to demonstrate that if you combine standard of care with the drugs that we're developing, that you would have a safe and effective way of prolonging survival time. I don't think anybody in cancer typically talks of cures necessarily. No. It depends upon the cancer indication, but certainly we'd be looking for a significant prolongation of survival time.
Brilliant. Is there anything you'd like to leave listeners with as a takeaway from everything we've been discussing today, whether it's from the ribosomes being worked with to the name Ribonexus? Is there a sort of one-sentence statement?
I think what we're trying to do at Ribonexus is to take something which is inherently very complex biology and turn that into something which can really have a significant impact on cancer patients. That is what Ribonexus is about.
Lovely. Well, thank you for your time, Stephen.
Thank you.
It's been a pleasure.
Thank you very much.
And so that concludes this episode of the Mini pharmaphorum podcast. You can find more information about this episode, including a download link and information about previous instalments and the main pharmaphorum podcast series at pharmaphorum.com/podcasts. The Mini pharmaphorum podcast is also available on iTunes, Spotify, Acast, Stitcher, and Podbean, where you can find and subscribe by searching for pharmaphorum. Of course, don't forget to visit our website, where you can sign up for daily news and analysis bulletins as well, and follow us on Twitter at pharmaphorum. Well, that's all for now. Thank you for listening.